Now we move on to our distinguished presenters from Israel to discuss how they've been battling the problem over there for the last several years. Our first presenter is Professor Stanley Freeman. He is with the Volcani Institute. He is a plant pathologist. He studied here at UC Riverside. In addition to avocados, he works with mangoes, strawberries, and other crops over in Israel, focusing a lot on soil-borne pathogens, but also on the pathogens that are related to pests like the shot-hole borer.
Good morning. Firstly, I'd like to thank Index Fresh and the California Avocado Commission for their generous support, which has allowed us to come here and share our experiences with you. And secondly, I'd like to thank Ken Melbourne from the California Avocado Commission for his warm hospitality, which has allowed our stay to be so comfortable. Firstly, I'd like to just point out that the complex that we are dealing with and all our experience until now is related to what you know as the complex appearing in the LA area. What is appearing in the San Diego area is a beetle complex which we're not familiar with because we only have the one single introduction. So I will be focusing more on the pathogen side of this complex, as I'm a plant pathologist and I work with fungal plant interactions, and my colleague Professor Mendel will be dealing with the beetle aspects. So just as an introduction to show you what varieties we grow in Israel, very similar to you probably, the Hass variety is our major cultivar, and not surprisingly, it's the most susceptible. So we have the other varieties which you're probably familiar with. This is the area that we grow in Israel. The expanse is about 7,000 hectares, actually probably more, close to 8. The phytopathological aspects which we are dealing with are similar, I think, to what you are dealing with. We have Phytophthora, although it's a minor pest or a minor problem in Israel, only located in certain specific areas. Rosalinia, which is another soil-borne pathogen, also only in certain areas. We have a Botryosphaeria complex, which is devastating our orchards at the moment. 2 species, which are different from what has been reported here in California. So I'm not sure whether our methods of control will be similar to how you would deal with it. But our fourth and, let's say, very damaging complex is the branch dieback associated with the ambrosia beetle that we've been dealing with, I think, for the last 5 odd years. And I'd like to share our experiences with you now with regard to that. So regarding the spread, our scale bar is 100 kilometers, which is about 55 miles. So you can see since 2010, this was when the first detection of the beetle fungus complex was recorded around the Tel Aviv area. We have avocado plantations in this area. 2 years later, it had spread all along the coast to the major avocado growing areas up north, also in the Bay Area of Haifa, and up to the northern border area where we have a lot of avocado growths. Also in Jerusalem area, we have a botanical garden, which in that area we detected the beetle on certain host species, so we were able to associate some of those botanical species to what we're growing in the garden here and look for the beetle in other trees growing in the urban areas. 2013, there was another jump, and a further spread of the beetle. And 2014, you can see a much larger encompassment of the areas that were infested. And that's probably where we are located at the moment, where we have an area down here which is clean, is not infested yet, and we have another area up here which is also not infested, but it'll just take it'll just be a matter of time before the beetle reaches these areas. Okay, as far as infestation goes and the levels of infestation, so we've surveyed about 22 farms encompassing about 20% of an area where infestation has occurred. So approximately half of that area is non-infected. We have serious infections as well, which are depicted in this picture, where you have huge limbs or mature limbs which are dying and have a lot of fruit drops, so we're losing yield as well. And the light infections are only just the initial secretion or excretion of the sugar compounds, the persitol, which show initial attack. Now, regarding the host range of plants which are infected, we have about 49 botanical species. Of those, there were 13 that the beetle tried to attack. We never found any fungal symbiote, or it wasn't really looked at because they were mock inoculations, mock attack. Here we have about 5 of those 49 which showed that the beetle was not successful in colonization. You have to understand that the beetle feeds on the fungus. So if the fungus is not able to colonize the tree, then the beetle will not have any feed to allow it to progress and complete its life cycle. So certain hosts which react and don't allow the fungus to colonize are, for example, olive and an ornamental tree called brachychiton. We have additional hosts which are attacked, and the fungal symbionts are isolated, but they aren't able to successfully colonize the xylem. So in that respect, the beetle cannot establish itself and complete a life cycle. However, of those 49 species that we've looked at, 15 so far are very suitable and allow reproduction of the beetle. So in that respect, you see symptoms and you see very serious branch dieback. In some instances, we will also get mortality of trees, but at the moment we're not seeing mortality of avocado. We're seeing mortality of oak, of box elder, and castor bean. Okay, just to show you some of the symptoms which you may have seen if you visited the southern part of California. So these are typical reactions of the tree that has been attacked by the beetle. You find the secretion of the persitol, the sugar, and serious infection. You'll have branch dieback. You'll also find through the season a lot of fruit that falls from those limbs which have been affected, so there is yield loss. And when you cut open those infected limbs which are infected, you'll see the typical galleries or tunnels which the beetle has excavated within the xylem, and you'll also see the fungus which lines the xylem and is the feed for those beetles, including the larvae which hatch from the eggs. I'd just like to point out that young trees are also infected. So we need to also be aware of this stage of a tree, but the tree has to be a woody tree. In other words, you're only talking about from the year 3.5, 4 and onwards. Young seedlings in the nursery are less susceptible, and I'll show you some data as we progress. In castor bean, these are typical symptoms where you see the pockmarks of the beetle penetration. Again, you see the infected xylem with the gallery as opposed to healthy xylem. And in oak, again, you have the gumming, which is similar to the sugar which was secreted in avocado. You have gumming in oak trees, which are also a reaction to infestation. And here you can see the typical browning or staining of the internal tissues due to the presence of the beetle and the fungus, and I'll elaborate regarding the staining as we progress. Again here, typically necrotic tissue within the xylem of the oak, and here you have penetration holes from the beetle. Box elder is very, very susceptible, and here we have dead and dying trees which we have to eliminate and remove. So as you can see, this is a process which is ongoing, and all these trees have to be removed because they are a severe source of beetles that can reinfest. Now, just to point out, persimmon is a tree which seems to be tolerant. The beetle attempts to infest and attack, but the fungus cannot colonize and establish itself, so this is considered a non-host. There is gumming. I'll relate to this as we progress as well. Olive trees, for example, are also mock attacked. You see attempted penetration here by the beetles, but when you peel away the bark, you can see that the beetle hasn't managed to penetrate because the fungus has not established itself. Okay, now regarding the fungi that we have isolated in Israel associated to our ambrosia beetle, we have 3 different fungi. They are identical to the ones reported by Dr. Escalen from UCR, and we are working very closely with Akif. So the first fungus that is detected, and it's detected within the heads of the beetle carries the fungus or the fungi, these symbionts, very actively and not passively on their bodies, but within certain structures within the head, and Professor Mendel will elaborate on that in his talk. The second fungus, which has been identified as a Graphium species, and the third, which Tim related to as well, is Fusarium uwolacea, which appears to be one of the major fungi involved in this complex. As you can see, the beetle, the larvae will ingest the mycelium of these different fungi, and the beetles and larvae feed on the Fusarium, on the Graphium, but do not seem to produce a complete lifestyle when grown on Acremonium, so we're not sure exactly what the role of Acremonium is, but the other 2 fungi seem to support a full life cycle of the beetle. Just to show you what these fungi look like up close, you can culture them, they're easy to manipulate. This is Acremonium, this is what it looks like microscopically. The Graphium is a darker looking fungus, and these are the spores, much smaller spores. And the Fusarium was also able to be cultured in plates, and the spores are much larger. Now, regarding the temperature of optimal growth of Fusarium, I'd just like to show you this graph that shows that the optimal growth is around 25, and the limits of growth are from, say, 10 degrees to 35 degrees, talking about Celsius. So this is not a limitation to the spread of the fungus. So the fungus is not limited in avocado orchards, so there are no restrictions to the growth of the feed then of these fungi when they have been introduced to the trees. Looking at the genetic diversity of the Fusarium population, I just show you one gel. I won't go further into this aspect of research, but we isolated Fusarium from many sources. We have over 200 isolates from different species of urban trees, from the beetles themselves, and when you do a genetic comparison between these different isolates, what we found out is that the Israeli and the California LA Fusarium complex are identical, and this is just to show you a pattern of amplification of certain genes. So these are the Israeli isolates, and here you have two representatives from California, so they're identical, as opposed to two other populations of Fusarium. So it's very easy to differentiate between these populations based on some of these molecular tools. So I won't go into this further in any depth. So just to summarize to this point, the Fusarium population in Israel appears to be uniform. It seems to be a clonal population, which indicates we had one introduction. Obviously, this is different from what has been reported in California, where in the LA County area you have the identical complex that we have. However, you have an additional complex with a different Fusarium beetle population around the San Diego area. So I'd like to go into the association of the different symbionts with the beetles, and again we're talking about the three different fungal species and how they interact with the beetles at each different stage of development. So here again you have growth of Fusarium and growth of Graphium, so they can grow together in Petri dishes. And this is just to show you that you can isolate these different fungal species from the heads of these beetles. This is an intermediate stage beetle. This is an adult stage beetle. They're usually differentiated according to the color of the outside of them. And here you have a mat of Fusarium populations, very high populations isolated from the heads of mature beetles. So just to take this one step further, we're looking at the different growth stages of these different beetles. We have the young stage, which is we term callow stage. We have an intermediate stage, which is a browner stage, and the black stage, which is a mature stage. And we're looking at the different proportions of each beetle within the heads of fungi, within the heads of each beetle at different stages. And we started looking at the populations within avocado. So this is what you see. You see low populations at the callow or early stages of mature beetles. As they mature further, the populations of Fusarium seem to increase, and at the mature stage we are finding mainly Fusarium. So we're not sure what this means, but it seems that Fusarium is a very important source of feed for the beetle, where at the mature stage it will carry its feed with it when it moves from place to place. The pattern seems to, or the trend seems to be very similar when you look at the beetle population, the fungal populations in castor bean. And again, as the beetles mature, the majority of fungi detected within the heads are Fusarium. And similarly, in box elder, the pattern is quite similar. Okay, so we wanted to look to see whether it's possible to, or let me just summarize that in a pie shape. It's much easier if you look at it this way. So at the callow stage, you have the majority of fungi being Graphium at the very early development stage. At the intermediate stage, the population seems to change, and we see much more Fusaria. And at the adult stage, the majority of populations within the heads of the beetles are the Fusaria. In experimental trials, by raising the beetle on different feed, we used to start off with Graphium, and it was very interesting because you can grow the two fungi together, the Graphium and the Fusarium. They don't interact, and there's no antagonism between them. So we were able to raise beetles artificially on Graphium, and at the mature stage, they indeed harbored or host or had within their heads the majority of fungi being Graphium. We're not sure exactly how they picked up the Acremonium and the Fusarium, but if they're artificially reared on Graphium, then the majority of population is Graphium. And this is opposed to those beetles collected in nature that possess only or nearly 100% Fusarium. So again, this is a question that we need to address. What the significance of each fungal species on the beetle is in nature as opposed to the proportion when the beetles are grown in the lab. Okay, turning now from the interaction between the beetles and the fungal symbionts to what exactly happens in the nurseries when you try and inoculate young plants with the Fusarium. So this is really of concern, I think, to the nurserymen, and these experiments were conducted on plants of approximately a year to a year and a half. These are seedlings of Hass grafted on rootstock. So these are the experiments, the experimental plants. As you can see, after about 10 days inoculation, here we have a water control as opposed to a Fusarium inoculation. You can see the reaction is very massive with the secretion of the sugars. Here's a close-up. 6 months later, we were very surprised that the plants remained healthy and there was any detrimental effect of the fungus, and they were inoculated at distances of maybe 3 to 4 inches along the stems. There was no detrimental effect to the inoculated young plants in comparison to the controls, which were water inoculated. Now, looking closely, when you section these stems, you can see there is a bit of staining within the tissue of the Fusarium inoculated plants, but they still remained healthy. And when you actually section those plants, you can see that the Fusarium was very, very localized. In other words, there was no systemic infection and the fungus did not progress throughout the stems. These are the controls. You can see the injury from wounding, and here you have the injury from wounding, including Fusarium, but again, very localized to where the puncture holes occurred. We also inoculated shoots and stems of young plants to see whether there was any difference between the actual stems themselves to the shoots. So 30 days post-inoculation, here you can see again in controls the reaction of secretion of sugars, and here you have the fusarium. Again, you have a more pronounced reaction, but again, no detrimental effect on the development of these buds. The buds just broke and carried on growing without any effect with the fungus that was inoculated here. We also inoculated by wounding, and as you can see, 6 months post-inoculation, this was the control wound, which seemed to heal. And here we have, again, a very similar healing of the wound when the fungus was introduced through the actual puncturing of the stem. So again, there did not seem to be much of a detrimental effect to the inoculation of the fungus in these vegetative tissues. What I'd like to do is just to compare the mechanisms of attack between this fungus in young plants as compared to other fungi which we are dealing with and which we are aware of. And the first pathogen which I'd like to refer to is the laurel wilt fungus, the fungus called Rafflesia loricola, which appears in Florida. And what you can see here is there's a rapid wilting and death of these seedlings within about 30 days following inoculation. So here you have a completely different reaction of the seedlings inoculated with this pathogen from the other ambrosia beetle from Florida, as opposed to our Fusaria from our ambrosia beetle. And in addition, when you take our Botryosphaeria pathogens and you inoculate on seedlings, within 4 days we can get mortality of these seedlings. So this is a very, very aggressive fungal pathogen which causes mortality in seedlings very fast. Just to summarize what I just showed you, so you have the pathogen which I'll just mention first, our Fusarium pathogen from our ambrosia beetle. So looking at seedlings and looking at trees, you can see that we have only local infections and not systemic, so L refers to local. We have a protection of seedlings but not of mature trees, and wounding is required for penetration. If you look at the Rafflesia pathogen from the ambrosia beetle in Florida, you can see that you get systemic infection of seedlings and trees. There's no protection of either type of plant material, and wounding is required in both cases. Botryosphaeria, exactly the same. So the Botryosphaeria is also systemically transmitted within the xylem. There's no protection, and wounding is required for symptom development. Now moving on to the infection in woody tissues, the picture is completely different. Here you have infection from the beetles. You can see the penetration holes. However, further from or in different directions from the introduction of the beetle, you can see a staining and browning of tissue, and this tissue is non-functional. So we for sure see the damage which manifests itself in a dieback of these branches. Now, is the fungus present here or not? This is a question which we're trying to address. So this is to show you that after artificial inoculations in mature orchards, within 2 weeks we obviously see the reaction of the limbs from infection. we see there's a browning, staining of the internal tissue as the fungus establishes itself. And 1 year following inoculation, you can see again there's a staining which has spread. We were not sure what the staining, what the source of the staining is. Is the fungus present there? Is the fungus causing the staining, or is it a reaction of the plant itself? The questions were addressed in such a manner, or let me just summarize this before I go further on. The fusarium that we have in our area does not infect vegetative avocado tissues, and this was shown with the young seedlings. The fusarium causes symptoms in woody avocado tissue only, and the infections by fusarium are localized and they're not systemic as compared to the Rafflesia and the Botryosferia fungi affecting avocado. Now, where exactly is that fungus located within the xylem if it doesn't move systemically, and what does the browning or the staining indicate? So we have tried to address these questions by sampling different areas of infected limbs using a sampling of cores. We take out sections from different areas to try and detect the fungus in such a manner. These are the cores that are taken out from the different sections, and these cores are sectioned themselves and plated on media to detect where the fungus is present or if it's not present. So these are the different kinds of reactions that you can expect. We can expect, and these are just the legends. You have a stained tissue, you have healthy tissue, you'll have presence of the fungus or no fungus. So diagrammatically, this is what we can expect, and this is what we found out. So these represent different sampling points where the cores were taken out. And what was very interesting is that in the majority of the stained tissue, the fungus was not present. So it appears that the staining of these different tissues is not directly associated to the presence of the fungus, but perhaps the fungus is releasing some kind of effector or some kind of toxin, or something is moving throughout the stem, but the stem will be non-functional. So it could be also a reaction by the plant itself. Again, here you can have different examples. The majority of the brown tissue or the infected tissue would not necessarily be infected by the fungus, but although brown, the fungus is not necessarily present. This is a question that we are trying to address at the moment. Exactly what is causing this browning and non-functionality of this infected tissue. Again, just to show you some of these results that we achieved. So again, these are cores that were taken out of infected tissue. Here we have distances. So we would have a 5 centimeter, which is approximately 1.5 inches away from galleries. This is a gallery where the beetle was present. The fungus was also present, but 5 centimeters away, although you did find staining, you could not detect the fungus. Again, this just shows you that in the majority of cores that were taken out of these infected tissues and plated, we only found the fungus, or very rarely found the fungus, beyond the gallery parts. So the fungus seems to be very localized and does not move itself. Just to summarize, the fusarium was not detected systemically. In other words, it does not move throughout the xylem. The stained woody tissue is non-functional, and that leads to a branch dieback and wilting. And the fusarium was not always detected in the stained tissue, but it seemed to be restricted to just a few centimeters or inches beyond the gallery. So it does not move, as opposed to other fungi, the Rafaela, which causes the dieback associated with avocado in Florida. Now, we wanted to determine whether the bark of the tree itself perhaps protected the penetration of the fungus. In this case, we artificially inoculated avocado, and we took a non-host persimmon also to look at this. So this is just to show you how we artificially inoculated, removed the bark, made penetration holes, and inserted fungal spores into these penetration holes. We did similar experiments with persimmon, which is a non-host, and these were the reactions of the persimmon. It has a different kind of gumming reaction, and the sugars are not sugars in this case, as opposed to avocado. And to summarize those experiments, when you look at the avocado as opposed to persimmon, here you have different sampling points, presence of the fungus, absence of the fungus. So these are graphs depicting with bark or without bark in avocado. You can see that the fungus in this case does not need the bark, or the bark will not be a restriction. In persimmon, we had very less or decreased presence of the fungus, and also the bark did not seem to restrict here. So there seems to be something within the persimmon tree which is restricting the growth of the fungus, and therefore this could be a replacement crop in Israel. And we know of farmers which are replacing the avocado with persimmon, which seems to be a much more tolerant crop. Okay, so to summarize that, the bark did not restrict penetration. The fungus was restricted to areas where it was inoculated. It did not progress further. And the non-host persimmon restricted colonization of the fungus, even though the bark was removed. Now, taking it one step further to look at ways of perhaps controlling the fungus in the field, as Tim mentioned, pathologists usually start in the lab with a whole range of fungicides to see what you can control the fungus in plates initially, and what works in plates can be thereafter applied in the field. So we first expose the fungus in plates to fungicides that are amended in the medium, growth medium, and then you check to see how the fungus grows and at what pace, to see how perhaps the fungicide can inhibit the growth in plates, and maybe in the field the same reaction will take place. We also used a similar plate assay to look to see how insecticides may perhaps inhibit the beetles. This was also based on a quite elegant assay whereby we grew the fungus on different sources of pesticides and then introduced the beetles to the fungal mycelium that may have took up the pesticides, and the beetles, by eating this different amended fungicide, pesticides in the plates, which were absorbed by the fungus, would perhaps be poisoned by the pesticide uptake. So this was applied in the lab, and pesticides which seemed to be effective under lab conditions were thereafter applied in the field. So this will also be reported by Tzvi later on. Here you can see the beetles eating the different types of mycelium, which may have the pesticide absorbed to it if grown on these amended plates and absorbed within to the feed. So this is the underside of the plate, you can see galleries formed, which are very reminiscent of what actually takes place in the orchard itself. This is just to show you that there are some fungicides which are very effective in plates. For example, here the fungus has remained at its initial growth stage. It did not grow, as opposed to other fungicides which are much less effective and do not inhibit growth of the fungus. Now, I'd just like to mention that we chose our fungicides according to modes of action and what types of, whether they were systemic or whether they were local. And this is usually based on the FRAC grouping, or the fungicide resistance action committees. Each different type of fungicide has a different grouping, and committees usually are established to see how efficient they are and to see what types of mode of action each fungicide plays a role against the different fungi. So we looked at the benzimidazoles, which are from FRAC group 1. These, again, are all systemic, the lists of fungicides which I'll be talking about. They inhibit microtubule formation, which causes cell deformation if they are active against certain fungi. We used DMIs, which are sterile biosynthesis inhibitors, and they cause misfunction of membranes, these fungicides. We looked at the strobilurins, which are inhibitors of respiration. They're also systemic, some of them. And growth inhibition is FRAC group 9. So just to summarize, some of these fungicides in plates, here we have an insecticide just to show you that it was not effective, as opposed to similar to the control of non-amended medium. So there were many fungicides which were very effective. Here we have FRAC group 3s, prochloraz, tebuconazole. The carbendazim were also very effective. Again, this is only against fusarium. Some were less effective, as you can see here. This is at a concentration of 10 ppm parts per million. Looking at other groups of fungicides, the metconazole seems to be also very effective, similar to reports by Dr. Escalén from UCR. I'd just like to mention that this fungicide has been discontinued in Israel, so we cannot use it, similar to the propiconazole, which is available here in the US. So although some are not available, there are others that do seem to have an effect, but only in plates. And I'd like to elaborate, just to make a point and stress that point. The less toxic fungicides, which would be considered more environmentally friendly, did not seem to have much effect against the fungus per se in plates. Just like to mention this this compound, which is actually an insecticide and seemed to have quite a good effect against the fungus per se. So this had some kind of potential. Okay, now all those results pertained only to fusarium. So taking the best of the fungicides which we had screened against fusarium, we screened them against the graphium and acremonium, which are also complexes of fungi found in the galleries themselves. So this is to show you that certain fungicides were good and effective against all three fungi. However, not all of them were as effective when comparing, for example, prochloraz to tebuconazole. So when choosing a fungicide, you have to be certain that it's effective against all three symbionts if that's the avenue that you're going to be taking. Okay, So taking it one step further to field trials, unfortunately there's not much promise here, and I'll elaborate now. The methods of application that we implemented were injection via the drip irrigation. We extensively irrigate by drip in Israel. We don't have any other method of irrigation to save water. There's no sprinkler, or there's very little sprinkler irrigation. This was one method of application. We also used stem injections, as you can see here, with syringes, first drilling holes, then inserting the syringes under pressure with the different fungicides. Again, here at a sectorial type of injection to try and get the fungicides within the tree and to move. So, and a third method was by soil drench around the drip irrigation systems next to the trunk of the trees to try again to get the fungicides to move systemically. We didn't have any success, unfortunately, in all our field experiments. I think, and I'll mention some of the points which were limiting in our with the rates of success, and that's it was very difficult to monitor initial infection due to the fact that the fungus itself was not always present. So it was very difficult to know how your fungicide worked, if it worked or not. The evaluation for efficacy, again, was based on how the tree developed, and you could not always associate what was happening in the tree, whether it was just the fungus or the beetle or both the compounds, the complex itself that was causing the damage, and movement of fungicides. Very, very few fungicides move within the xylem of a tree, and they usually are absorbed. They do not move. So there was very, this was very disappointing, and we unfortunately never had any success when applying fungicides to naturally infected trees. Our future research, which we'll be carrying on, is based on the specificity of the beetle to botanical host to try and ascertain what are the types of hosts that are attracting the beetle, what types of hosts are repelling the beetle, and trying to ascertain what are these compounds that are involved. We'll be looking at specificity of the fungal attraction to the beetle. Some of the fungi are more or we assume, the hypothesis is that the beetle is attracted to some of those three symbionts in different manners. So we're trying to learn exactly what fungus is more attractive to the beetle, and perhaps manipulate the beetle in this manner, and to look at the colonization of the host tree itself, and to determine the dynamics of fungal colonization, to see exactly what that staining effect is and how to perhaps remedy the tree and to reduce that staining. So with that, I'd like to acknowledge the people that funded this work. That's the Avocado Growers Organization in Israel and the chief scientist of the Israeli Ministry of Agriculture. Thank you very much.
There are differences in the phosphite movement in avocados, depending on the time of year, the phenological stage of the tree. Was that taken into consideration in your fungicide applications?
Well, we're using the phosphites to try and boost the reaction of the tree to induce it to resistance against Botryosferia. We have extensive field trials, and I cannot relate so much to the movement as opposed to what's happening in the tree itself. When you inject, we tried injecting, we tried spraying against the ambrosia beetle and the fungus itself without any success. Against Botryosferia, yes, we do have some type of success. I cannot say much about movement because we aren't doing any extensive work at this stage to find out exactly how the tree is reacting, and we are only doing sprays, not in any injections. I don't know whether that answers your question, but
Have you looked at what mechanism that the olive or persimmon trees might be having that's inhibiting the growth of the fusarium, the fungus, as a way to?
no, but that is of extreme interest to us because the fungus is not establishing itself, and therefore there's no feed for the beetle. But no, we haven't, and that's one of our research objectives.